Well, hello, everyone. I'm really happy to be with you today. I'd like to thank the organizer of this conference, the International Society for Vaccines, for inviting me. It's a great pleasure to be with you. My name is Cyril, and as you can see from my slide, I'm the National Program Leader for the Animal Health National Program here at Agricultural Research Service, which is the research branch within the United States Department of Agriculture. And the title of my presentation is a research and development of veterinary vaccines from a biodefense and One Health perspective. Now, in the time that's been allowed to me, I'd like to cover the following topics. I know I'm going to have to go very fast, but I will do my best to bring the points that I want to make, but I apologize if I'm not able to cover all of the information that's all in the slides that I'm going to present with you today. So I'm going to start with a very brief introduction of pathogens of concerns. And I want to introduce the U.S. National Biodefense Strategy, then share with you why veterinary vaccines are important for biodefense. Then I'll share with you some of the challenges associated with veterinary vaccines. Then we'll very briefly cover new concepts and approaches for the future of vaccine development. And then I'll just go over two brief examples for two important diseases that I've selected for the purpose of this presentation, and that is foot and mouth disease and African swine fever. And then I'll have some concluding remarks. So I think most of you are probably familiar, at least those of you that are in the United States, with the Select Agent Program that's administered both by the CDC and the United States Department of Agriculture. That's a list of agents that are the most concerned, the most dangerous pathogens. And as you can see from those bullet points, Health and Human Services has their own list, agents like smallpox and anthrax and tularemia. The USDA has also their list, which includes foot and mouth disease and African swine fever. Then we have those overlap select agents, which are our zoonotic agents that are transmitted from animals to people, where the reservoirs are animals. And then we have these tier 1 select agents, which are the most dangerous, and that includes smallpox. And there's actually two agricultural agents on that list, one being rinderpest, which is the second infectious agent that's been eradicated from our planet thanks to human intervention after smallpox. And the other one is foot and mouth disease. Now, no matter how hard we try to have the list, and those on this list are certainly regulated because of the concerns associated with them, but it's also a struggle for other agencies and, in fact, government agencies throughout the world to really know what is going to be the most pathogen. Now, when you look at the USDA, we've got our own list of what we consider high consequence agents. As you can see from the title of their slide, those are foreign animal diseases and pests. We have tier 1 agents, again, African swine fever and classical swine fever, foot and mouth diseases, and avian influenza, not only the highly pathogenic strain, but the ones that have zoonotic significance. And in fact, I've highlighted in red font on the USDA list all of the agents that are zoonotic agents. But again, no matter how hard we try to prepare, we're always surprised, right? Because we have these emerging diseases. And I think most of you will agree that none of us were ever able to predict the emergence of the pathogens that are on this list. On the left side, I've listed several pathogens that you'll recognize. And then on the right side, animal pathogens. I've placed a star next to all the agents that are zoonotic agents, and as you can see, there's a number of them. And then also I'd like to introduce this concept of a reverse zoonosis, whereby it's people that can transmit an infectious agent to animals. And good examples of that are pandemic H1N1 in 2009, where swine workers actually infected swine. And currently with COVID and SARS-CoV-2, we've actually had cases of people infecting mink on mink farm. And that's of a concern because then we have a potential new source of virus that can spread and be part of the epidemiological picture in terms of a source of virus for people. So how do we prepare? Over the years, we've seen several documents, several strategies. One back in 2004 was the National Strategy for Countering Biological Threats. And some of you may be familiar with this one, where in 2017, Congress passed the National Defense Authorization Act to essentially establish a biodefense strategy that says that the Secretaries of Defense and Health and Human Services, Homeland Security, and the Secretary of Agriculture shall jointly develop a national biodefense strategy. That led in 2018 to the publication of the President's strategy, which essentially says that we recognize that there's a need for a collaborative, multi-sectorial, or OneHealth transdisciplinary approach to the national biodefense enterprise. And in the strategy, there's 5 strategic goals. And the one that is relevant to this conference is goal number 4, that says rapidly respond to limit the impact of bioincidents. And specifically looking at one of the strategic objectives, 4.2.3, it says that there's a need to conduct real-time clinical research to evaluate promising investigational medical countermeasures and identify potential novel diagnostics, treatments, and vaccines. And of course, when we think about COVID, that led to several task forces, one of which was Operation Warp Speed, that generated for the first time in history, in record time, SARS-CoV-2 vaccine that we've been able to deploy in record time to try to stop the spread of SARS-CoV-2 in our country and many other countries across the world. However, as we know, it wasn't exactly perfect. We know just by looking at the number of deaths. I mean, my goodness, today I think we have up to 620,000 American citizens that have lost their life due to this emerging pandemic, a tragedy. And so the question is, how can we do better? And the current administration, the Biden-Harris administration, is in the process of doing a whole government review of our national biopreparedness policies, which will be released later on this week. And later on this year. And just last week, they published this document, the American Pandemic Preparedness, Transforming Our Capability. This document essentially describes the work that is needed to transform U.S. capabilities to respond rapidly and effectively to any future pandemic. And this document lays out 5 pillars, 12 strategic objectives, and has identified a need for funding these strategic objectives and their implementation at $65.3 million. So here are the 5 pillars. I'll let you read them, but the one that's really relevant to this conference is transforming medical defenses, and specifically looking at vaccines. There's 5 strategic objectives, and even though this is geared specifically for human health, the goal, as you see listed, is really around how can we develop vaccines faster? What are the improvements that can be made around logistics, manufacturing, and distribution, right? So the first one, vaccine design. How can we develop vaccines that against any human virus within 100 days? Those are very ambitious goals, obviously. Vaccines for the entire United States population within 130 days, enable the delivery of vaccines rapidly and easily to anywhere in the world, and vaccine administration. And the last one is really a real challenge in the world of vaccine development and vaccinology. How can we develop ways to rapidly adapt, test and review and modify vaccines to keep pace with changes in the virus, as we've seen with the emergence of variants such as the SARS-CoV-2 Delta virus we're very concerned that if it continues to mutate, our vaccine may no longer be as effective as we need them to be. Now, I'm confident that later on soon, we will also identify specific needs for veterinary vaccines. And I will tell you that I will say ditto. I mean, most of what's been identified here for human vaccines is applicable to veterinary vaccine. In fact, I will say that for veterinary vaccine, in fact, we have a lot more needs, and we need these vaccines to do even more. And I'll cover that in a few minutes. But first, let's just go over very briefly on the importance of veterinary vaccines. So, one, we know that we're going to grow the world population by 2045 to some 9 billion people on this earth, and we're going to need to feed them. And I will tell you that animal agriculture is very important. In fact, there's over 1 billion people on this planet that are dependent on animal agriculture for their livelihood. And even countries like India that have more of a vegetarian-based food staple, that they actually have the largest dairy industry, because they convert the milk into edible protein, which is very important for a healthy diet. And then we have the challenge of these new and emerging diseases. In fact, 70% of these new emerging diseases are either vector-borne or zoonotic. So for veterinary vaccines, we really need vaccines for production diseases to mitigate economic and public health threat, and vaccines for rapid real-time response whenever we face a disease outbreak. And really, we need to have more of a global view of these emerging diseases and pathogens, so that we can have a better approach to the global control and the eradication of these diseases. In fact, if you look at documents such as this one that was published first in 2010, as this recently been updated in 2017, this document was produced by the Food and Agriculture Organization of the United Nations, along with the OIE, or the World Organization for Animal Health, and the World Health Organization, or the so-called tripartite. And they've committed to essentially working together to improve our ability to respond to animal and public health risk attributable to zoonoses and animal diseases with an impact on food security. In fact, the concept behind this is simply that we probably can do a lot better by controlling these diseases at the source, by preventing them from spilling over into human populations in the first place. And in this diagram published in 2012 by the World Bank, they're trying to make that point by looking at it from an economic standpoint. And as you can see, the economic impact of zoonotic disease outbreak goes up exponentially as you wait, and as the virus spreads in animal populations and spills over into human populations. So let's move on to look at challenges for veterinary vaccines. So like human vaccines, we're missing a lot of vaccines for important diseases such as African swine fever. We need vaccines for highly mutable RNA viruses. Vector-borne diseases continue to cause a significant challenge in veterinary medicine, and we really need vaccine for those as well. Like SARS-CoV-2, we have a number of emerging diseases, including coronaviruses that we've experienced in the United States, such as porcine epidemic diarrhea virus, for instance, that came from China. So we really have a need for vaccines that are going to be effective against these emerging pathogens. I've touched base on the need and the potential impact of having vaccine for zoonotic diseases. But what we really need is vaccines that have now been engineered specifically for the control and the eradication of these diseases. So when we look at these vaccines and ask the question, what do we need them to do? Well, here's an important list. One is we need these vaccines in veterinary medicine to work with one shot, right? We can't wait for a vaccine to to induce a protective immune response where it requires 2 or 3 vaccination. We need a rapid onset of protection. This is really important when we face a disease outbreak. We want to stop that transmission. And so the sooner we can induce a protective immune response, the sooner we can prevent that disease from spreading. For a lot of these agents, like influenza viruses and even foot and mouth disease, where we have a lot of different strains, we need vaccines that are efficacious against all strains, because even if we are aware of these pathogens, we never know what strain is going to emerge and come to our shores. An important concept of veterinary medicine is vaccines that have been engineered to differentiate infected from vaccinated animals. We call that the DIVA strategy, DIVA. That's really important for recovery, because if we implement a vaccine campaign, we want to make sure that we know which animals have been vaccinated, and the animals that have been vaccinated and exposed are in fact not spreading the virus. That's important, especially in agriculture, when we need to get back to trade. And of course, we need vaccines that are cost-effective, scalable, and delivery systems that are enabled, that will enable mass vaccination. Now, this is a plug for the research and research priorities. I apologize, I don't have time to go through all of the research priorities, but just looking at the first 3, to enable the profile that I just described in the previous slide, we really need to understand mechanisms of pathogen-host interactions, such as the mechanisms that these pathogens use to evade the immune system. What are the primary sites of viral replication? If we want to stop transmission, then we need to induce a protective immune response at those sites. Understanding how we can engineer vaccine to have the highest protective immune response. And going back to vaccines that work with one shot and that work rapidly, we really need to understand mechanisms of onset of immunity, for instance. So at my agency, we're really focusing on a lot of this basic research to try to engineer these vaccines. So very briefly, new concepts and approaches. Sure, I don't have to explain to this group that's listening to this presentation today, this new technology that first was presented by Rep. Oli, this reverse vaccinology concept. Another important tool is this reverse genetic systems that have been developed for RNA viruses, such as depicted on this slide for influenza viruses. And then today we have all these different vaccine platforms that I've listed here, including the messenger RNAs vaccine platforms that were used to rapidly develop the SARS-CoV vaccine. Now, in veterinary medicine, I will tell you that we've probably historically in the last 20 years been more advanced than on the human side in the development and use of these vaccine platforms, not because we're any smarter than our colleagues on the human health side, but strictly because we can test directly the safety and efficacy profile of these vaccines directly in the relevant host. But a word of warning, right? Because we have to be careful about technology for technology's sake, because what we really are trying to get at here is, what platform is actually going to help us solve the problem. So just because, we have messenger RNA vaccine platform now, that's not going to work for a large majority of the vaccines that we're trying to develop for important diseases such as African swine fever, for instance, a very large vaccines with multiple genes where we really don't know the function of over half of these genes. So, the question would be then, what genes would we clone in this messenger RNA? So it's very different than some of the low-hanging fruits that we have, such as influenza viruses and coronaviruses, where we know the protective antigen, right? Coronaviruses, the spike protein, and of course, we know that we can develop an effective vaccine against influenza viruses by having the right hemagglutinin gene product. So what I'd like to share with you is that we really need to move from old to new approaches. So move from 19th century vaccinology, or Pasteur's rule of vaccinology, where he told us that we could isolate, inactivate, and inject and have an effective vaccine. But there the goal was really to prevent clinical disease. And although we've had many successes with that, we have many diseases, as I explained, that really pose many challenges. And so we really need to move to more of a systems biology approach, using some of the latest tools that we have in research, either molecular virology, functional genomics, immunology, and looking at the transcriptome, the proteome, and even the metabolome. The goal here, and the point that I'm trying to share with you, is that when we look at these vaccines, again, we really need to look at what do we need to engineer them to do? What do we need these vaccines to do to make sure that they're going to be effective in the control and where feasible eradication of diseases? So it's really all about problem solving, and to steal a term that's used by scientists that develop diagnostics, we really need these vaccines to be fit for purpose. So very briefly, almost running out of time, so I just want to share with you two examples. The first one is foot and mouth disease. The vaccine platform here that I want to share with you uses reverse genetics. This was a vaccine developed by Elizabeth Reeder and Luis Rodriguez on Plum Island. The name of the vaccine is DFMD Lethallus 3B3D. And the problems that we were trying to solve for foot and mouth disease is simply the ability to manufacture a vaccine for foot and mouth disease on the US mainland. Many of you know that this is a tier 1 agent, as I mentioned, and no one is allowed by law of working with this agent on the US mainland. And so we cannot manufacture vaccines on the US mainland, and that provides delays whereby we have to source vaccines from foreign countries. And in fact, historically, there's been disease outbreaks that have been caused by vaccine virus escaping from manufacturing plants. And other problems that we're trying to solve is the ability to rapidly develop those vaccines. FMD vaccine, as I previously mentioned, have several strains, 7 serotypes, some 60 subtypes, so you have to have the right vaccine strain, or else your vaccine is not going to be effective. And we need to be able to implement a robust DIVA strategy. So very quickly, what you see here is the sequence or the genome of the FMD virus. And what Elizabeth Reeder and her team did was essentially take the backbone of an FMD virus, the 824, a vaccine virus that was already, a virus that was already adapted to the cell line that's used to produce FMD vaccine. And Elizabeth deleted the leader sequence, and which is involved with evading some of the interferon pathways, and essentially totally attenuate this virus, so that if that vaccine virus, when it's grown for production purposes, if it escapes, then it really doesn't matter, because it will not cause any disease in the target animals, either cattle or pigs. And what she also did here, if you go from the left to the right, when you look under the structural area, the P1 region, 1A, 1B, 1C, 1D, she inserted 2 restriction enzyme whereby you can swap the cassette that codes for the genes for the virion, what the immune response sees for any virus, for any strain, and then inserted 2 negative markers in a non-structural proteins, 3B and 3D. So now we have a vaccine that was specifically engineered to better help us respond and control a foot and mouth disease outbreak if it ever occurred in the United States. I'm just about out of time, so very quickly, the last example that I wanted to present for you was an African swine fever vaccine. And, excuse me. And this vaccine is a live gene-deleted vaccine. It uses a reverse vaccinology approach, and the problems that we're trying to solve is the fact, as I alluded to, that we have a virus that after 50 years of research, we've never successfully developed a vaccine, in large part because we don't know the function of over 50% of this very large virus. Our first approach, our first generation vaccine is going to be a live African swine fever vaccine. But historically, we failed to produce a safe vaccine or even one that was efficacious. And so what our scientists on Plum Island did, specifically Manuel Borca and Doug Gladue on Plum Island, is they created a custom computational pipeline to take into account different sources of bioinformatics information and functional genomics data. And with that, identified new African swine fever protein targets. And they identified this gene, the I177L, that is predicted to have immune modulatory, to be an immune modulatory protein. And by deleting this gene and replacing it with a fluorescent marker, DM cherry, we essentially were able to test this vaccine and identify that, in fact, this one I177L is a determinant of virulence. And this candidate vaccine is, in fact, very efficacious at a very low dose. It lacks total virulence when you inoculate pigs at a high dose. There's no vaccine shedding, and it induces, at least in our studies, sterile immunity, in that we can't recover the challenged virus in the target tissues. So, by the way, both of this vaccine and the Lilo-7D vaccine are currently have been transferred to commercial partners and are currently being developed for a purpose. So very quickly, conclusions. I have 4 of them. I hope I've been able to convince you of the importance of veterinary vaccine for biodefense. Second, scientific discovery is delivering promising new vaccine candidate tools and technologies to take on seemingly intractable infectious diseases. 3, technical advances are providing new research tools and opportunities that are providing scientists a hitherto really unprecedented ability to engineer vaccines that are fit for purpose. And lastly, there's really a need to establish animal health research laboratories here in the United States and across the world that are dedicated to the development of veterinary medical countermeasures to support the progressive global control and, where feasible, eradication of these transboundary and emerging infectious diseases. In fact, I'd like to introduce that USDA will very soon have the National Bio and Agro Defense Facility, a $1.4 billion facility that will, for the first time, give us the ability to have large animal rooms, not only against BSL-3 agents, but BSL-4 agents, some of the most dangerous pathogens. And this facility will also have a biological development module that will allow us to do some early development and facilitate the transfer of our technologies to industry for their rapid development. So with that, I'd like to thank you very much for your attention. It's been a pleasure to be with you and had the opportunity to present this presentation. If anyone has any questions for me, please don't hesitate to contact me at cyril.gay@usda.gov. Thank you very much for your time and attention.